as a result of this. Is it clear? One more time I will explain, because this is really important part. You could see the arrangement of the rod cells. Many rod cells are converging to one point. Around roughly, you could say 4 or 5 of them will converge to one point, and that point will be the beginning of bipolar neuron. And the bipolar neuron will end in a region where there will be the beginning of the cell body of the optic neuron. And many optic neurons will form optic nerve. And this arrangement is so special because when you look, you can see the outer segment arrangement and the inner segment arrangement. When you call outer, we will be thinking that the arrangement is in the opposite direction, but no. The cones and the rods, outer segment is facing the region of the retina, whereas the inner segment is facing towards the bipolar neuron. So as a result of this unexpected arrangement will make all the neurons to be converging to one point. And that point of the primary neurons ending, we call it as the optic nerve. When they all have to pass through one layer, we call that spot as the blind spot. As you see in the picture of figure D, there is no light sensitive cells in the blind spot. That's why if the vision falls there, we won't be able to see anything because there is no light sensitive cells. Now let's be familiar with the structure of cone. Have a look at the structure of cone. The shape of cone, outer segment, is like cone. That's how it got the name cone cells. Similarly, outer segment of the cone cell also is the light sensitive region, which also contain flattened membranous vesicle. But over here, it's not visual purple, but we call that as, over here, the pigment is called iodopsin. Tell me, type and tell me what is the name of the pigment of the rod cells which we discussed. Yes? Yes, correct.
Rhodopsin?
We call rhodopsin. You can type, that's better, no need to talk. Okay, fine. Yeah, most of the people are typing as rhodopsin, visual purple, correct. So that means rods are having rhodopsin, whereas cones are having iodopsin. But structurally, they have similar structure. They also have, both of them have outer segment, both of them have inner segment. And since they both are specialized cells, they both have nucleus and synaptic region, everything, the structure is same except the pigment. Now, we have to learn something very important. That's why I have sent that picture. The picture in which the working of the rod cell is being shown. Please check the picture which I have sent you, okay? Now we will be learning from your book figure E in page 254 and then go back to the picture that I have sent. Both are very clear for the explanation of bleaching and dark adaptation. Often this can be addressed in the exam also, which means how do the rod cells work or how do the cone cells work? To explain that, we need to have an idea about the structure. I hope the structure of the rods are clear, right? Now, we will talk a little bit about how the reaction of rhodopsin towards light. Please check figure E. Figure E shows that a photon of light is hitting the rhodopsin. Rhodopsin is the light sensitive pigment. The moment light hits on rhodopsin, as I told you before, there is a chemical breakdown. And this chemical breakdown we call as bleaching, the change of shape, that is from the cis-retinal to the trans-retinal form. That happens, and this will create a generator potential, which we have learned that, for example, we will not call it action potential since it is not a neuron, it is a secondary receptor. In all the secondary receptors, we will be calling it as the generation potential, right? So this created a generator potential, and the size of the generator potential is very important. It depends upon the amount of light hitting the rod, and the bleaching will also be with the light. And this generator potential, when it reaches the threshold, we get the action potential. And that we will not say depolarization, instead we say generator potential. This action potential the nerve impulse will be carrying along the optic nerve to the brain. And that's how we see in dim light. Not only we can see in dim light, we can check out the slight movements in the visual field also in low light intensity, because these sensory receptors are very, very sensitive to even the lowest intensity of the light, or you could say it can work in dim light. This is because of it. Now, to explain this in more detail, we will conclude the process. What happens if it is dark or after a long time of exposure to light? Why do we blink? Because in normal daylight, the rods are almost completely bleached. So that's why with every few minutes interval, we will be blinking. And this blinking will make dark adaptation, which means rhodopsin will be completely reformed, which means, see the arrow, rhodopsin is resynthesized in dark condition. This means when you blink, when you are in a bright sunny exposed area, after some time you will be closing your eyes. That 1 second blinking will help the retinal and opsin to get resynthesized to form rhodopsin. And I told you before, there is a lot of mitochondria in the inner segment. This mitochondria will help in joining of retinal plus opsin. That ATP is required there so that rhodopsin is resynthesized. That means it's back to the, in dark it will be cis-retinal and in light it will be transretinal. Is this idea okay? You can type it if it is clear. Then I'll go to little more detail of this. Yeah, okay. Little more details of this from the picture I have sent. Can you look into the picture which I have sent you just now? It says dim light and it says dark, right? Okay, so let's talk about in dark what is happening. Just to concentrate on the rod cell. We have learned before that the neurons will not allow the sodium ions. That means neurons are impermeable to sodium ions. But in the case of the receptor cells, which means if you are talking about sensory receptors, if you are talking about the secondary receptor, in this case, it is a rod cell. Sodium ion is very much permeable. This has nothing to do like neurons impermeable. This is really permeable, which means that the rod cells will normally allow the sodium ion to enter inside. So when you keep on checking the picture I sent you, it says that sodium ions continuously pumped out. Can you see it? And pumped in, pumped out, pumped in. This process is keeping on going. As a result of this, if you trace the dark adaptation, we are going to learn about the dark adaptation now, which means that there is always sodium movement. This influx of sodium movement will slightly depolarize the cell. In this condition, the depolarization is not minus 70, it is minus 40. That means which will cause the release of a neurotransmitter substance. This neurotransmitter substance is said to be called glutamate. And when glutamate is synthesized, glutamate will cause no release of neurotransmitter from the bipolar cell. When bipolar cell is not releasing any neurotransmitter, this creates no action potential. And if there is no action potential in the optic nerve or optic neurons, many of them will be called as optic nerve, in that case, you will not be seeing anything. That's why at night or where there is no light at all, there is no visibility. When you talk about the other half, you will understand more clearly. Imagine their photon of light or light falls on the retina. When light falls on the retina, just now we told it is bleaching, which means that the rhodopsin is breaking down into retinal and opsin. Now, opsin will be functioning as an enzyme and it will undergo a lot of series of changes. As a result of these cyclic, or you can say the cascade reactions, there is closure of the sodium channels or sodium ion channels on the outer segment will be closed, which means the influx of sodium ions is blocked, but sodium will be continuously pumped out of the inner segment. Outer segment is blocked, but inner segment sodium will be continuously pumped out. When more and more sodium is pumped out, this creates an hyperpolarization condition inside the cell. When it is hyperpolarized inside the cell, you can see that no transmitter substance is released, written. So if you write no transmitter substance released, which means the bipolar neuron can become at that time depolarized. When the bipolar neuron is depolarized, it will release a excitatory neurotransmitter substance, which means that the action potential is generated along the optic neuron. So what I want you to understand is both the sides, the dim light and the dark light. And if you think this picture is not clear, exactly the same thing is mentioned in your textbook. But in your textbook, they did not write about the sodium part with the picture. That's why I have sent that picture. But basically the idea is same. Action potential will be generated along the bipolar neuron. If action potential is generated on the bipolar neuron only, it will release a neurotransmitter substance, which can cause an action potential on the optic nerve, right? So this is the idea which happens only in dim light. Is that okay? So we need in between darkness also, or blinking of your eye. Why we need blinking of your eye? So that we can resynthesize rhodopsin. If rhodopsin is not resynthesized, we won't be able to view again in bright light. I want you to go home, and today, oh, you're at home only, sorry. I want you to do 255 page, the checkpoint today. Then only you will understand all the ideas more clearly. I suppose we can move to habituation. Yeah, some people are saying yes, fine, okay. So can you go to the next page, 256, habituation. Yes, since we have learned about the junction and the neurotransmitter release and the depolarization on the postsynaptic membrane, habituation will be easy for you to understand. What do you exactly mean by habituation? Habituation means it's a learning process. In this learning process, almost all animals, including human, is also involved. You know that when there is a big sound, when a baby is really, really small, the baby will be just throwing back the baby itself, its arms, and grasping. This moment, the baby is startled, and this is what we call it as the baby is alarmed. Why the baby is alarmed like this? Because the baby is not habituated. Slowly, 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 the baby will learn. So habituation is a learning process. The baby will learn, and the baby will not be acting anymore like this. The baby will become slowly, when the baby grows and matures, it understands that this is a response which we should learn and practice, right? So that habituation is a learning process in which how we are adapted to one condition. That is what we are going to learn. How is this really happening? This is really happening with the help of calcium ion channels becoming less responsive. So we will explain the process, okay? And then we will go back to one example with giant African land snails. We can study a small investigation with habituation. A lot of examples can be given, but you need to know how to respond to that. For example, how habituation is being understood by an animal and how the animal learn to ignore this stimuli, whatever you are giving, if it is just a normal stimuli. If every time, if it is keeping on acting to that stimuli, that means the animal is responding. When the animal is not habituated, and if the animal is all the time responding, animal will be wasting a lot of its energy for that. So the animal understood that it is not a dangerous situation, or it can be an avoidable situation. So this response is in the memory. So this is a crucial element in learning. And that's why habituation is involved in brain development. And for human, we can say how memories are formed, how this has been helping the human in responding. For example, go to figure B. Figure B, you can see how a small animal is being habituated with water jet, and it understood that it need to ignore the stimuli. For example, the animal is being introduced with water jet. So when the animal is getting this stimuli for the first time, the animal is not habituated. So the animal will be quickly withdrawing its gills when the siphon is stimulated. You can see the picture of the siphon and you can see the picture of the gills withdrawn. After several minutes, if you again repeat the stimuli, the animal will keep on doing it. But repeatedly, if you are giving the same stimuli, the animal will become habituated to this. This is a sea slug. Slowly, slowly, the animal will understand the stimuli and the animal will stop responding to the stimuli. How the animal will stop responding to the stimuli? With repeated stimulation, calcium channels become less responsive. What do you mean by that? That means less calcium will enter inside the presynaptic neuron. As a result, less neurotransmitter vesicle will be moving down the presynaptic node and less neurotransmitters will be released. If less neurotransmitters are released, only less neurotransmitters can diffuse through the synaptic cleft and can attach to the postsynaptic receptor. If less neurotransmitters are attached, obviously less protein gates will be open. If less protein gates are open, this means slowly, slowly the depolarization in the postsynaptic membrane slows down, which means no action potential will be triggered on the motor neuron. So thus, the vesicle movement will be less and the depolarization will be less. Thus, the action potential will be less. And finally, there will be no response at all. So this condition is said to be called as habituation. So habituation is a behavior pattern. Frequent exposure to the same stimuli, whichever animal it is, the animal will learn that. And that's a very easy example which made the scientists to study the nervous system's performance. We can do this experiment of habituation with small animals like snails, and we can understand that if you touch with a small earbud, wet earbud, if you touch on the tentacles, the tentacle will be withdrawn. The animal will keep on doing it for few times, and afterwards the animal will not even respond. This make you understand that the animal is habituated. Gentle touch will do, the animal will respond. So it's very easy to understand. Is the idea of habituation clear? Okay. Have a look at the central nervous system. Go to page 262. Central nervous system consists of the brain and the spinal cord. You know that. Of which both of them are important in controlling a lot of coordinated responses depending upon the situation. Obviously, you know that the nerve fibers in the spinal cord will help in all unconscious reflex actions. But how is a brain formed? You see the picture that I have sent. It's a very nice picture where the neural tube of the embryo is shown with the forebrain, the midbrain, and the hindbrain. And at the end, the remainder of the tube will form the spinal cord. In your book, it is written central canal. From the central canal, that tube will more fold and make up a long structure, which we call as the spine. The formation of the brain in vertebrates, the brain is very simple, and we can see different sites which coordinate with 100,000 million neurons. And when you look into the structure of the brain, the picture that I have sent, we can see the cerebrum itself with many different layers. We can see the frontal lobe, we can see the temporal lobe, the parietal lobe, the occipital lobe, like that, many different portions. Check the cerebrum only. The cerebrum, the left side and the right side of the cerebrum is connected by lots and lots of bands of axons of many different neurons, which we call as corpus callosum. Moreover, these hemispheres have different sites for different centers. In your textbook, the picture is not that good. That's why the picture which I sent, you can see the blue color part, the frontal lobe. The frontal lobe is for thinking, reasoning, decision making, and also it has lot of neural connection with the spinal cord. It gives information to the spinal cord. Just the region below that, you could see olfactory. That means it is only related to smell. A region below, you can see the green colored part. That is the temporal lobe, which will help in hearing and then sound recognition, speech, understanding the speech, and then the memory part. A little above the green part, you can see a yellow colored part, which is said to be called the parietal lobe. This is associated with the memory, recognition, calculation, sensation, all those. And the violet colored part is called the occipital lobe. This is one of the most important part which we are learning now, which will help in visual sensory area. That means everything related to eye. What you see, how you interpret what you are seeing. These are all the parts of the cerebrum. So once again, what are the parts of the cerebrum we learned? Frontal lobe, temporal lobe, parietal lobe, occipital lobe. These are the lobes. Then followed by, you can see the cerebellum. The cerebellum will help in posturing, balance, and motor coordinations and all those. Then you can see the pituitary gland, and we have learned about the pituitary gland. So you know more details about the pituitary gland. Then comes two small swellings. The first swelling we call as the medulla, which will control the breathing rate, the heart rate, blood pressure, and all of them. Below that, you can see pons, that is concerned with the breathing and also the heartbeat coordination. Similar like the medulla. And then you can see the hypothalamus. Hypothalamus is little above the pituitary gland. Thermoregulation, osmoregulation, thirst, hunger, aggression. Also, you have learned the coordination with some hormones. So you know it, right? Now we will speak about, and I want you to do one thing. You will be making a table. That is the easiest way to learn. Make a table and write each part of the brain and then write the functions of each part of the brain. Like for example, if I'm asking one role of medulla, can you type what is the role of medulla? Any one role of medulla. Yes, like that. If I ask one role of hypothalamus, yes. So they will be most of the time asking this, and so I want you to have a table, and inside the table put all these and learn. If you are asked to describe brain and its functions, you should be able to. And there's an exam hint given. Remember that the brain activity relies on conduction of action potential and synaptic transmission. Memory relies on forming particular nervous pathway connection between the cells. So all those things you need to be a little bit clear about. Now we will talk about reflex action. You have already learned what is a reflex, so it is easy for you to know. What are reflex? Yes, actions without conscious thought. We call it as reflex. Most of the time, below the head, the actions are coordinated by the spinal cord. And above the head, it is the head region will be definitely coordinated with the brain only. And the spinal cord is a very important coordination part. Blinking, coordination, dilating the pupil, swallowing, hand moving. These are all some unconscious reflex examples. Now we will talk about the reflex action with the reflex arc in detail. I want you to go to page 264. Page 264. Yes. In page 264, you can see the spinal reflex. This picture, figure C, is very, very important. The spinal cord and the structures involved in spinal reflexes. I'm sure that you all know this, but few portions we are not clear about. For example, you know the pathway. For example, you have learned that every time a stimuli will be received by a sensory receptor. Sensory receptor. I repeat, it is not a nerve cell. It's a sensory receptor. Just now we have learned about two sensory receptors, which are photoreceptors, and we learned them as rods and cones. And you remember last class we learned a lot of sensory receptors. For example, we learned proprioceptors, chemoreceptors, mechanicoreceptors, photoreceptors, thermoreceptors. So this one could be a thermoreceptor, a sensory receptor, a thermoreceptor. That means it's a sensory cell which is in the skin, which can be for the temperature sensing. And that part we need to understand. And then keep on tracing the pathway. This sensory receptor is giving the impulse to the sensory neuron. This part you know it, it's a neuron, yeah. And the sensory neuron carry the impulse. You could see a small swelling, and that swelling is a ganglion, where there is the cell body of the sensory nerve fiber, and through that it moves and enters to the gray matter, and this time this is the gray matter of the spinal cord, where it relays the information to the junction, which we call as the synapse. From the synapse it is taken by the neuron, which we call as the relay neuron, and the relay neuron will give this information to another junction, which we call as the synapse. From the synapse, it will be again carried by another neuron, which we call as the motor neuron, and the motor neuron fibers from the spinal cord. It is moving and ending in an effector, and that effector will be either a muscle or a gland. But what you don't know now, the whole pathway you know it, but what you don't know is the ganglion. So you have to be familiar with the ganglion and the dorsal root part and the ventral root part. That means if they give this picture, you have to describe the reflex arc and you have to annotate it fully, which means you have to write about it and you have to speak about each part of the picture. So you know that the stimulus will be the pathway. If you want to explain the spinal reflex, you can tell that any of the sensory receptor that will be receiving the stimuli, and then this impulse will be traveling through the sensory neuron, and it will, the pathway you will describe, and it will end in the gray matter of the spinal cord, where it will be ending into the synapse. From that synapse, it will be taken by the relay neuron, and then it will enter to the next synapse, where it will be taken by the motor neuron. And motor neuron, this part you don't know, so you will emphasize this. The movement was through the dorsal root, and the coming out part will be through the ventral root, and it travels through the effector, and the effector is most commonly a muscle, and that muscle will respond. Responding is by contracting and pushing away the hand, or pushing away whichever part it was been exposed to. The muscle contraction will lead to that. So please go through the spinal cord structures and the spinal reflexes and be familiar with that. Now, the next page, the size of the pupil. This light causing the pupil reflex. Very well you have learned in IGs, but we will put a lot more different ideas into it, which you haven't learned. For example, you have only learned about the iris muscles, the circular muscles and the radial muscles, what happens to that, right? But we did not bring the cranial nerves in action, which means this reflex, we call it as a cranial reflex. What do you mean by cranial reflex? Which means the relays between the brain and the parts of the head. It could be to the neck, or it could be to the eye region, any part of the brain. When we are discussing that reflex, we will call it as the cranial reflex. So in this case, we will be talking about, as you have learned, the muscles in the iris. But that's not enough. The muscles in the iris, the control centers of these nerve impulses will be with the parasympathetic and the sympathetic cranial nerves, which means we are going to talk about ocular motor. Ocular motor is the real name given for the eye and the neurons associated with it. So in this case, when we are talking about the basic principles of the pupil reflex, you will just involve the sympathetic and the parasympathetic part also. That's the only one part which you haven't learned in the IGs, which you will be talking this time more importantly. So let's learn the simple way, and then we will involve the parasympathetic and sympathetic to it. For example, if there is dim light, obviously you know that the pupil will become wider. When dim light is there, why the pupil is becoming wider? So that it can accumulate more light into it. But how the pupil is becoming wider? Did you know that this reflex is controlled by both the eye, even if light is with one eye only? That means if bright light is shown to one eye, but the narrowing of the pupil will be done by both the eyes. If dim light is shown to one eye, the widening of the pupil is also done by both the eyes. I'm sure that you might not have thought about it, right? Why is it so? We will see that picture and come back to the dim light. What is the principle of the pupil reflex? I want you to go to figure E. Figure E in page 265. The cranial reflexes that control the dilation and contraction of the pupil of your eye are much more complex than the simpler spinal reflex arc. We are not talking about the spinal reflex, but it is more complex, the cranial reflexes. As you can see, the changes in the size of the pupil more easily when you closely observe, you see both the eyes over there. You check the bright light coming in. The picture, the bright light is shown only to one eye, but the reflex is being exhibited by both the eyes. How is that? You trace the pathway. The sensory receptors taking the information, sensory neurons taking the information to the optic nerve. But when it comes to the control centers in the brain, have a look at the picture. You can see from the left side, it's coordinated to the right side. From the right side, it is coordinated to the left side, which means the control centers in the brain will take it to the motor neuron of both the right and left part. And from there to the relay neuron of both the right and the left part. The relay to the motor, that is the ocular motor nerve, and then the information will come to the effector on both sides, which means that if bright light shown only on the right side of the eye, still the left will get the information, and the circular muscles of the iris in the left and the right, both of them will constrict, and the radial muscles in both of the eye will relax. Thus, if a bright light is shown on one eye, both the eye, the pupil will become narrow. Even if you were closing one eye, it's an involuntary reflex. If you close right eye and a bright light is shown only to your left eye, right eye will also respond. If you close left eye, instead if you close right eye, also the left eye will respond. Close right eye, both the eyes will respond. Close left eye, both the eyes will respond. Which means the control centers in the brain will take the information to both sides, right side and left side. Now let's see what is the basic principle of pupil reflex. The basic principle of pupil reflex is very easy to memorize. We start with dim light. You can think that dim light, that is D. In dim light, circular muscle will relax. DCR, RC. DCR, RC. DCR, RC. DCR meaning dim light, circular muscle relax, radial muscle contract. And P for pupil. Pupil will become widen, PW. Just learn one of this, then the whole idea will become clearer. I'll tell you what happens. When the light is shown on, let's say we'll talk about the dim light, yeah? Dim light enters into the eye. When dim light enters the eye, what should happen? The pupil should be more wider. Why should be the pupil more wider? So that more light can enter and you can see things. The opposite should happen in bright light. When bright light comes in, very little pupil should only be open. Otherwise, too much bright light entering with too much frequency of action potential, then it can cause damage to the photosensitive cells of the retina. So that's why the pupil will become constricted. How is the pupil constricting? The circular muscle contract. BCC, bright light, circular muscle contract. RR, radial muscle will relax. And PW, PN we can say. That means the pupil will become narrower. this is the idea which you already know. But now we will tell one more thing. The control centers in the brain, which will send nerve impulses, has a branch in the parasympathetic cranial nerve. That means when you are talking about bright light, you will talk about parasympathetic cranial nerves. That means which will allow